Hey guys, my name's Max and welcome to Simple Biology. In this video we're going to keep talking about cellular respiration, and in this video in particular we're going to talk about the citric acid cycle and the electron transport chain. Now this is our second respiration video, so if you haven't actually seen the first one, please go back and watch that one first because a lot of things we're going to be talking about won't make sense unless you've seen it in order. Now the citric acid cycle and the electron transport chain are actually where the cell gets a majority of its energy when we talk about respiration. In fact, I think it gets like 18 or 19 times as much energy as in glycolysis. So obviously, these two phases are going to be very important, especially for eukaryotic organisms, which actually have this phase of respiration. But before we actually get into all the processes of these two phases, there's an important molecule called acetyl COA that we're going to have to talk about before we get to there. Now, essentially, COA is what's called a coenzyme. It's coenzyme A. Now, a coenzyme is basically any organic molecule that acts like an enzyme, but it's not actually a protein. So it'll catalyze some sort of reaction, and it's organic, but it's not a protein. In this case, really all you need to know about it is that it has this sulfur at the end. And what will happen is it'll, in one of these steps, it'll bond to a carbon. And this weak bond that's represented by a wavy line is a very important aspect of acetyl COA, and that's all you really need to know about that coenzyme. Essentially, there's a series of steps that that are used to transfer or to convert pyruvate into acetyl COA. And those steps are what we're going to be talking about on this slide. The first step is that a carbon dioxide molecule is formed. You can see right here that there's the CO and O in the pyruvate, and they're basically just taken off to form carbon dioxide. Next, an NADH is formed. So this NAD plus is oxidized using hydrogens from the pyruvate, or from that intermediate after pyruvate. And then a coenzyme A is actually attached to the remainder, making it less stable. This whole process is actually fueled energy-wise by taking off this carbon dioxide. Taking off the carbon dioxide will actually release energy, and that energy will be used to reduce NAD plus and to attach coenzyme A. Now, this acetyl COA is actually very unstable. It can easily react with other compounds, and that's the function of that coenzyme A, is to make it unstable. And so now that it's unstable, it can go into the citric acid cycle, which is what we're going to be talking about next. Now, the citric acid cycle is, I guess like the name suggests, a big cycle where you have molecules that go in, and as the cycle goes on, you eventually use those molecules again, or those atoms within the molecules at least, to form the compounds, and it just goes around in circles. It's also known as the tricarboxylic acid cycle, or the Krebs cycle. Now, the citric acid, which is a very important molecule in the cycle, is what's known as a tricarboxylic acid because it has 3 carboxyl groups. And if you remember back from our chemistry unit, the carboxyl group that citric acid has is an acidic functional group. So that's why it's called a tricarboxylic acid. And it's also known as the Krebs cycle for the guy who actually kind of did a lot of the work in figuring out how it works. His last name was Krebs. You don't really need to know that, but what you do need to know is that the citric acid cycle will take place in the mitochondria. So in contrast with glycolysis, which occurs in the cytoplasm, this is where the molecules will actually go into the mitochondria in order to react in the citric acid cycle. The net gains, as far as the citric acid cycle goes, are 3 NADH, you can see right here, 1 FADH2, which we'll talk about in just a second, and a gain of 1 GTP or ATP right here. It also produces 2 carbon dioxide. Now, FADH2 is very similar to NADH. It's an electron carrier, but it will actually take 2 hydrogens, where NADH will only take 1. They essentially play the same role, but slightly different, and they're different molecules. They're actually different molecules, so they'll do the same thing, but with slightly different results. GTP is actually this very similar compound to ATP. It's a guanosine triphosphate instead of adenosine triphosphate, and the only real difference is a little nitrogenous base on the end. And oftentimes the GTP will actually be used to form ATP, so you really don't need to worry about it at all. Now, these NADH and FADH2 have to actually be used by the cell in order to get any energy, and so that's where the electron transport chain comes in. Basically, what will happen is you'll have NADH2 and FADH, or NADH and FADH2 that will be oxidized in order to give their electrons to these different proteins in this electron transport chain. And each protein is actually more electronegative than the last. So every time an electron is transferred along this line between these proteins, energy is released, and that energy is where the energy comes from in respiration. There are four major proteins that we're going to talk about, and they're actually numbered 1, 2, 3, and 4, and they're located on the inner mitochondrial membrane. Like I said, NADH and FADH2 are oxidized in order to give their electrons to these proteins, and what they actually do with this energy is they pump hydrogen ion into the inner membrane space that's between the two membranes of the mitochondria. Now, if you remember, mitochondria, of course, have two membranes, and in that little space between them, that's where we have the intermembrane space. So since hydrogen ions are actually being pumped into that intermembrane space, you get a very acidic environment, and that acidic environment will want to neutralize with the basic environment in the center of the mitochondria. That's where the energy will come from in order to produce ATP. We'll be talking about that in our next slide. Now, like I said, this next step will actually use the energy from that separation of hydrogen ions in order to produce ATP. So what you have is this big multi-protein complex called ATP synthase. And you can see that there's a bunch of different proteins, and each one actually plays its own role. But as a whole, what will happen is these hydrogen ions will actually diffuse across the membrane. But as they do so, it'll force this top part to spin. It'll do this kind of in a similar manner to water in a water mill. And like how a water mill will actually produce something usable, sometimes energy, sometimes other things, this will actually produce something usable, ATP. That process is also called chemiosmosis because of the diffusion of these ions, or the proton motive force because these hydrogen ions are really just protons. And like I said, this spinning motion is what will drive the synthesis of ATP. What's also interesting is that this is actually the smallest rotor found in nature. And a rotor is basically anything that can spin in order to produce energy. And I just think it's interesting that there is no rotor smaller than this, and it works just in the same way that other rotors work. Now also, what you have to know is that this process will actually produce 32 or 34 ATP molecules per molecule of glucose, which actually represents 40% of the energy from that glucose stored in these ATP. Now 40% may not seem like a lot, but it's actually an incredible amount when you compare it to some other engines and other things that are much less efficient. So it's kind of interesting that these biological molecules are just so incredibly efficient. And really, the citric acid cycle and the electron transport chain are just as simple as that.